my name is Sofia Cruz.
Hi, my name is Ileana, and our project is on the effect of aloe vera edible coating on bananas.
The reason as to why we chose this topic was to be able to find new and efficient ways to preserve the shelf life of fruits and vegetables, in this case specifically bananas.
Several studies have tested aloe vera gel as the main source in helping fruits and vegetables last longer. Bioplastic edible food coating has helped various fruits and vegetables have a longer shelf life. This study aims to determine the efficiency of aloe vera gel used as edible coating on retaining the nutrient content in bananas. Bananas are known for going bad after about a week. We wanted to test our theory in this experiment.
The bananas that we chose for this experiment are the ones one would usually see at a grocery store, which are the Musa, acuminata, Cavendish bananas. Like my partner Ileana just mentioned, the bananas do not have a long shelf life, which results in them only lasting on an average of 5 to 7 days. We started this experiment by collecting a total of 40 bananas. Each of us had 20 and individually conducted the experiment in our own separate homes. 10 bananas for both of our trials were the control group, and the other 10 were the experimental group. The aloe vera gel was collected from an aloe vera plant in my backyard. It was sliced, and once the gel was collected, I placed it in a Ziploc bag and put it in a refrigerator overnight while Ileana blended it and placed it in a container and also left it overnight in a refrigerator.
The following day of collecting the aloe vera gel, we measured the girth, weight, and length of each banana. These measurements were collected every day, every 12 hours, one at 9 a.m. and one at 9 p.m., for a span of 9 days. We would also make sure to take a picture of our bananas each time we would collect the measurements for a way to capture the progress. The time length of the experiment all depended on how long it would be until the bananas would turn a dark brown color. When placing the aloe vera gel coating on the bananas, I coated the experimental group evenly and did 2 layers, while Sofia placed a non-blended coating directly onto the banana and also did a double coating. After coating, the separate groups are transferred on a tray and placed in a room temperature location. Throughout the experiment, physical chemical properties such as weight loss, color, firmness, and decaying were being recorded. After the 9th day, we collected the data, averaged it out, and used ANOVA 2-factor with replication statistical analysis to determine whether or not there was a significant change between the controlled and experimental group and overall efficiency of the gel coating.
As you can see on our poster in figure 1, that was the first day of my trial, and the bananas were a bright yellow. And on the figure 2, it was on the last day, the 9th day, and you can see a difference in the color in which the bananas turned a more dark yellow and did have a lot of brown spots and dark spots.
In figure 3, these are my bananas from my experiment on the first day, and they are a bright green, bright yellow color. And in figure 4, these are my bananas on the last day, on the 9th day, and you can tell that the color changed drastically. And figure 5 and 6, it shows the materials that we used, and we set it up with a notepad and a scale.
In table 1, that was where we collected the average pre-weight and post-weight of the bananas. The blue color indicates the pre-weight of the bananas, and the red color indicates the post-weight. On the left side, you can see that it was the experimental group of the bananas and the control group on the right side. And there was, as you can see, there was a demonstration of a difference in the weight.
On table 2, we recorded the average pre-girth and post-girth bananas, and the blue column shows the pre-girth and the red shows the post-girth, and on the left, we recorded it in centimeters.
And then on the last table, table 3, you can see that the average it was the pre-length and the post-length of the bananas. And just like the past tables, the blue indicates the pre-length and the red indicates the post-length. And on the left side, it's the experimental group, and on the right side, it's the control group. And there was a difference in the pre-length and post-weight and length. So after analyzing our data, the results demonstrated that there was a p-value of 0.04. This means that there was indeed a statistical significance between the control and experimental group. We noticed several differences between both of our trials. My trial demonstrated a loss of firmness for the bananas, and all of them were very close to a black color, which means that they were decaying.
My trial, on the other hand, did not have a drastic change in color compared to Sofia's trial. The bananas I used were still yellow with brown and black spots, and by the end of the experiment, they were still edible with minimal decay.
Our research showed that the aloe vera gel coating had a significant impact on the banana's shelf life. The average shelf life for a banana is about 5 days. The aloe vera gel was able to preserve the bananas for approximately 9 days. However, our research had limitations due to the COVID-19 pandemic. The research had to be conducted within our own homes. Methodology for both trials were different from one another. Materials needed to produce a more effective batch of aloe vera gel coating were missing due to the unavailability. Future studies could enhance the aloe vera gel coating by adding solutions to make the aloe vera gel more stronger.
We would now like to acknowledge Dr. Nora Garza,
Rosemary Agüero Riojas,
Leslie Solis,
Luisa Villarreal,
Gabriela Solis Cavazos,
and the Leaves Title III Award. Thank you, and thank you for watching our video.